Here we have the inactive site with the receptor, G protein, enzyme, and plasma membrane. Then a signal molecule, also called the ligand, enters the receptor and activates the G protein with addition to GTP. Then the G protein moves across the cell and activates the enzyme. The enzyme releases some type of cellular response as well as the G protein and phosphate group.
So the signal molecule is going to start to come in from outside of the cell, and it's going to dock in the signal binding site. And then once they're docked, the receptor tyrosine kinase proteins are going to start to move closer together to form the dimer. So then once they're connected, an ATP is going to come in, and then it's going to phosphorylize the dimer, and it's going to become a fully activated receptor tyrosine kinase. And then after that, the relay proteins are going to come in. Then they're going to connect to the phosphates and become activated relay proteins. And then those two will form cellular response 1 and 2. So in transcription, there's going to be a signal molecule, which is cortisol, that is going to come in from outside of the cell. And you can see the receptor chaperone complex cannot enter the nucleus because it can't. So the cortisol is going to come into the cytoplasm and it's going to bind to the receptor. And then the receptor is going to change shape and it's going to release the chaperone protein. And this is going to allow the receptor and the cortisol ligand to enter the nucleus, where they're going to go to the DNA, and then after that they can be transcribed.
A signal molecule attaches to a receptor but cannot cross the cell membrane, so it sends a message to attach to the first inactive protein kinase, causing it to become active. Then a phosphate is taken from ATP and hydrolyzes into ADP. Then the phosphate attaches to an inactive protein kinase, which causes a shape change to make the protein kinase 2 active. Then it catalyzes the transfer of phosphate off ATP to another ADP, and then transfers the phosphate to inactive protein kinase 3 and changes its shape to active. Then, when it's the end of the phosphorylation cascade, there is an inactive protein. Protein kinase 3 transfers phosphate group, and the protein becomes active. Finally, a cell response is produced.